Reduction of structures. In some cases, evolution has led to the reduction or loss of certain structures or traits. This occurs when those traits are no longer necessary or beneficial in a particular environment. For example, many cave-dwelling organisms have lost their eyes due to the absence of light in their habitats. Increase in brain size. Over evolutionary time, there has been a general trend towards larger brain size in certain lineages. This increase in brain size is often associated with enhanced cognitive abilities and higher levels of intelligence. Primates, including humans, exhibit this trend, with larger brains compared to their body size. Evolution of flight. The evolution of flight is a significant trend observed in several groups of organisms, including birds, bats, and insects. The development of wings and the ability to fly have allowed these organisms to explore new habitats, access food resources, and escape predators. Flight has led to the diversification and success of these groups. Increase in body size. In many evolutionary lineages, there has been a trend towards larger body size over time. This is known as Cope's rule, which suggests that within a lineage, organisms tend to evolve towards larger sizes. Larger body size can provide advantages such as increased strength, competitive advantage, or better thermoregulation. Coevolution. Coevolution is a trend that involves the reciprocal evolutionary changes between interacting species. For example, the arms race between predators and prey, where predators develop new hunting strategies and prey develop defenses against predation, is a classic example of coevolution. Evolution of novelty. Evolutionary trends also include the emergence of novel traits or structures that provide a selective advantage. These innovations can lead to the diversification and success of new lineages. For example, the evolution of jaws in vertebrates was a significant innovation that allowed for more efficient feeding and led to the radiation of jawed vertebrates. It is important to note that evolutionary trends are not universal and may vary across different lineages and ecosystems. Additionally, trends can be influenced by various factors, including environmental changes, selective pressures, and genetic variation. Nonetheless, studying evolutionary trends provides valuable insights into the processes and patterns of evolution over time. Biochemical evolution. Collapse of orthogenesis in detail. With respect to evolution, with examples of zoology in detail. ChatGPT. Biochemical evolution, also known as chemical evolution or prebiotic evolution, refers to the processes by which complex organic molecules, including the building blocks of life, arose from similar chemical compounds on early Earth. It played a crucial role in the origin of life and the subsequent diversification of biological systems. One concept associated with biochemical evolution is the collapse of orthogenesis, which refers to the rejection of the idea that evolution is directed towards a predetermined goal or follows a linear path of continuous improvement. Orthogenesis, or straight-line evolution, was a hypothesis popular in the late 19th and early 20th centuries. It proposed that evolutionary changes occur in a linear and progressive manner, guided by internal factors or predetermined forces. According to this idea, species would evolve in a specific direction, leading to a predictable and continuous improvement of traits over time. However, the collapse of orthogenesis occurred as accumulating evidence from various scientific fields, including paleontology, genetics, and molecular biology, challenged the notion of a predetermined evolutionary trajectory. The collapse of orthogenesis was significant in shaping our understanding of evolution, including in zoology. It led to the acceptance of the concept of natural selection, proposed by Charles Darwin, as the primary mechanism driving evolutionary change. Natural selection is a process by which heritable variations that provide a survival or reproductive advantage become more common in a population over successive generations. It operates on the variation present within a population, favoring traits that increase an organism's fitness in its specific environment. Examples of the collapse of orthogenesis and the dominance of natural selection can be observed in various zoological phenomena. Adaptive radiation. Adaptive radiation refers to the rapid diversification of a single ancestral species into a wide array of different species that occupy various ecological niches. The Galapagos finches, studied by Darwin, exemplify adaptive radiation. These finches evolve different beak sizes and shapes, allowing them to exploit different food sources on the different islands of the archipelago. Convergent evolution. Convergent evolution occurs when unrelated species independently evolve similar traits or adaptations due to similar selective pressures. It demonstrates that evolution does not follow a predetermined path but is influenced by the environment. For instance, the streamlined bodies of dolphins and sharks are an example of convergent evolution, where different lineages have independently evolved similar body shapes to adapt to the aquatic environment. Evolution of antibiotic resistance. Bacteria provide another example of the collapse of orthogenesis. The evolution of antibiotic resistance in bacteria is a consequence of natural selection acting on genetic variation within populations. Bacteria that possess genetic mutations or acquire resistance genes through horizontal gene transfer can survive exposure to antibiotics, leading to the emergence of resistant strains. Evolution of flight. The evolution of flight in different lineages, such as birds, bats, and insects, demonstrates the independent evolution of similar adaptations in response to the selective pressures associated with aerial locomotion. The wings of birds and bats, despite their different anatomical structures, serve similar functions, illustrating the nonlinear nature of evolution. Overall, the collapse of orthogenesis marked a paradigm shift in our understanding of evolution, highlighting the importance of natural selection and the role of environmental factors in shaping the diversity of life. It emphasized the idea that evolution is a dynamic and unpredictable process, driven by interactions between organisms and their environments. The study of biochemical evolution and the collapse of orthogenesis have deepened our understanding of the mechanisms and patterns of evolution, contributing to the field of zoology and our knowledge of the diversity and complexity of life on Earth. Stages in primate evolution including Homo, dry and wet-nosed primates. Prosimians and simians, human and the African apes, in detail. Big essay with multiple examples and major points in detail. ChatGPT. Primates, including humans, are a diverse group of mammals that share common ancestry and exhibit a wide range of anatomical, physiological, and behavioral characteristics. The evolution of primates can be traced back over 65 million years, and it encompasses various stages and significant developments. Let's explore the stages in primate evolution, including Homo, and highlight the major points and examples along the way. Prosimians. Prosimians are the most primitive group of primates, which include lemurs, lorises, and tarsiers. They exhibit several characteristics that distinguish them from other primates. Prosimians have a more olfactory-oriented sense of smell, and they possess a wet nose, characterized by a moist rhinarium. They also have a greater reliance on their sense of smell compared to later primate groups. Additionally, they typically have more primitive dental structures and exhibit a wider range of locomotor adaptations, including leaping and vertical clinging and leaping. Example, lemurs, found in Madagascar, represent one of the most diverse groups of prosimians. They showcase a variety of adaptations in ecological niches, ranging from the small, nocturnal mouse lemurs to the larger, diurnal ring-tailed lemurs. Anthropoids. Anthropoids are a group of primates that includes monkeys, apes, and humans. They are further divided into two major subgroups, New World monkeys and Old World monkeys and apes. Anthropoids have several distinguishing features, including a larger brain size, forward-facing eyes that provide binocular vision, and a greater reliance on vision rather than olfaction. A. New World monkeys. New World monkeys are found in Central and South America. They possess a flat, broad nose, often referred to as a platyrrhine nose, which differentiates them from Old World monkeys and apes. They also have a prehensile tail, which enables them to grasp objects and use it as an additional limb. Example, the spider monkey is a New World monkey known for its prehensile tail and exceptional agility in the tree canopy. B. Old World monkeys and apes. Old World monkeys and apes are found in Africa and Asia. They have a narrow, downward-pointing nose, known as a catarrhine nose. Unlike New World monkeys, they lack a prehensile tail. The apes, including orangutans, gorillas, and chimpanzees, are characterized by their larger body size and more complex social behavior compared to monkeys. Example, the chimpanzee, one of our closest living relatives, exhibits remarkable intelligence, tool use, and complex social structures. Hominins. Hominins are a subgroup of primates that includes humans and our close extinct relatives. The evolution of hominins is characterized by several key developments, including bipedalism and increased brain size. A. Australopithecines. Australopithecines are early hominins that lived between 4 and 2 million years ago. They are known for their bipedal locomotion, evidenced by the structure of their pelvic bones and leg bones. Australopithecines had a combination of ape-like and human-like features and had a brain size smaller than modern humans. Example, Australopithecus afarensis, represented by the famous fossil, Lucy, is an important example of an early hominin exhibiting bipedal locomotion. B. Genus Homo. The genus Homo includes modern humans, Homo sapiens, and several extinct species that evolved after the Australopithecines. The genus Homo is characterized by increasing brain size, refined tool use, and cultural advancements. Example, Homo erect. Continuation. ChatGPT. Continuation. Example, Homo erectus, an extinct species, represents an important stage in human evolution. They had a larger brain size than their ancestors and were the first hominins to leave Africa and colonize different parts of the world. They also developed more sophisticated stone tools and likely had complex social structures. C. Neanderthals and Denisovans. Neanderthals and Denisovans are extinct hominin species closely related to modern humans. They existed in different regions of the world and shared a common ancestor with Homo sapiens. These species had robust bodies and adapted to cold environments. Genetic studies have shown that modern humans carry traces of Neanderthal and Denisovan DNA, indicating interbreeding between these species. Example, the discovery of the Neanderthal remains in the Neander Valley, Germany, provided crucial insights into our evolutionary history and the existence of closely related human species. D. Modern humans, Homo sapiens. Modern humans, Homo sapiens, are the only surviving species of the genus Homo. They originated in Africa approximately 200,000 years ago and eventually spread across the globe, replacing other hominin species. Modern humans possess unique features such as a large brain capacity, complex language and culture, and the ability to engage in abstract thinking and technological advancements. Example, the fossil record of early Homo sapiens, such as the Homo inheritus skulls found in Africa, provides evidence for the anatomical characteristics of modern humans and their migration patterns. In summary, the stages in primate evolution, including Homo, trace the gradual development of primates from ancestral forms to the emergence of modern humans. From prosimians to anthropoids, including New World monkeys and Old World monkeys and apes, each stage exhibits unique characteristics and adaptations. The evolution of hominins, including Australopithecines and the genus Homo, highlights the progression towards bipedalism, increased brain size, and cultural advancements. Finally, the emergence of modern humans represents the pinnacle of primate evolution with our unique cognitive abilities, language, and global dispersion. The study of these stages and examples in zoology provides valuable insights into the complexity and diversity of our evolutionary history. Continuation. ChatGPT. Continuation. E. African apes, chimpanzees, and gorillas. Chimpanzees and gorillas are the closest living relatives to humans. They belong to the family Hominidae and share a common ancestor with humans. These species exhibit both physical and behavioral similarities to humans, such as complex social structures, tool use, and communication systems. Example, the study of chimpanzees, particularly the observation of their tool use and cultural behaviors, provides insights into the potential behaviors and cognitive abilities of our common ancestors. F. Human evolution. The evolutionary history of humans, specifically the genus Homo, is marked by several key stages and important species. These include Homo habilis, Homo erectus, Homo neanderthalensis, and Homo sapiens. Each species represents a significant milestone in human evolution, with distinct anatomical, behavioral, and cultural characteristics. Example, the discovery of the fossil remains of Lucy, an Australopithecus afarensis, in Ethiopia in 1974, shed light on the early stages of human evolution and our bipedal locomotion. G. Cultural evolution. In addition to biological evolution, humans have undergone significant cultural evolution. This includes the development of language, tool use, agriculture, and the establishment of complex social structures. Cultural evolution has played a crucial role in shaping human societies and enabling our species to adapt to diverse environments. Example, the transition from hunting and gathering to agriculture, which occurred independently in different regions of the world, represents a significant cultural milestone in human history and led to the development of settled communities and the rise of civilizations. Overall, the stages in primate evolution, including Homo, provide a comprehensive understanding of the gradual development of primates, culminating in the emergence of modern humans. Through the study of fossil evidence, comparative anatomy, genetics, and cultural advancements, researchers have pieced together the complex evolutionary history of our species. This knowledge not only enhances our understanding of our own origins but also contributes to our understanding of the broader field of zoology and the diversity of life on Earth. African origin 4. Modern humans, in detail with respect to evolution with examples of zoology in detail. ChatGPT. The African origin of modern humans, known as the out-of-Africa theory or the recent African origin hypothesis, is a widely accepted scientific explanation for the evolutionary history of Homo sapiens. This theory proposes that modern humans originated in Africa and subsequently migrated and replaced earlier hominin populations in other parts of the world. Evidence supporting the African origin of modern humans comes from multiple sources, including fossil remains, genetic studies, and archaeological evidence. Here are some key points regarding the African origin of modern humans. Fossil evidence. Fossil discoveries in Africa, particularly in regions such as East Africa and South Africa, have provided significant evidence for human evolution. Fossils of early hominine species, such as Australopithecus and early Homo, have been found in Africa, indicating the presence of our evolutionary ancestors on the continent. Example, discovery of fossil remains of early Homo. Example, the discovery of fossil remains of early Homo species, such as Homo habilis and Homo erectus, in East Africa provides evidence of human evolution and suggests Africa as the place of origin for our lineage. Genetic studies. Genetic studies, particularly those focused on analyzing mitochondrial DNA, mtDNA, and Y-chromosomal DNA, have provided compelling evidence for the African origin of modern humans. These studies show that modern humans have the greatest genetic diversity in Africa, indicating a longer period of evolution and genetic divergence on the continent. Example, analysis of mtDNA in various populations around the world has revealed that all modern humans have a common female ancestor, often referred to as mitochondrial Eve, who likely lived in Africa around 200,000 years ago. Genetic variation within African populations. Studies of genetic variation within African populations have revealed higher levels of genetic diversity compared to populations outside of Africa. This suggests that modern humans have been evolving in Africa for a longer period of time, accumulating genetic variations and adaptations to local environments. Example, the study of the genetic diversity among different African populations, such as the San people of southern Africa or the Hadza people of Tanzania, provides insights into the complex evolutionary history of our species within the African continent. Migration out of Africa. Genetic and archaeological evidence supports the idea that anatomically modern humans migrated out of Africa and populated other parts of the world. This migration is believed to have occurred in multiple waves, with different populations spreading across different regions. Example, genetic studies comparing the genomes of modern non-African populations with African populations have revealed genetic signatures of migration and interbreeding events, such as the presence of Neanderthal and Denisovan DNA in non-African populations. Overall, the evidence from fossils, genetic studies, and archaeological findings strongly supports the African origin of modern humans. It suggests that our species emerged in Africa, underwent various stages of evolution and diversification, and eventually spread across the globe, replacing or assimilating with other hominine populations along the way. The African origin theory is a cornerstone of our understanding of human evolution and provides insights into the evolutionary history of not just our species but also the entire animal kingdom. Y-chromosome atom and mitochondrial Eve. In detail with respect to evolution with examples of zoology in detail. ChatGPT. Y-chromosome atom and mitochondrial Eve are the hypothetical common ancestors of all living humans in terms of their respective genetic lineages. While they did not exist simultaneously, they represent the most recent common male and female ancestors from whom all modern humans can trace their genetic lineage. Here are the details of Y-chromosome atom and mitochondrial Eve. Y-chromosome atom. Y-chromosome atom is the name given to the most recent common ancestor of all living males in terms of their patrilineal descent. This means that every living man can trace his Y-chromosome lineage back to this individual. Y-chromosome atom is estimated to have lived between 100,000 and 200,000 years ago. Explanation. Y-chromosome atom is identified by studying the Y-chromosome, a sex-determining chromosome passed down exclusively from fathers to sons. By analyzing the genetic markers on the Y-chromosome across different populations, scientists can trace the shared ancestry and identify the most recent common male ancestor. Example. A study conducted in 1995 analyzed the Y-chromosome variation in different male populations and estimated that Y-chromosome atom lived around 140,000 years ago. However, it's important to note that this estimate is subject to change as more data and improved analytical techniques become available. Mitochondrial Eve. Mitochondrial Eve is a name given to the most recent common matrilineal ancestor of all living humans. This means that every person can trace their mitochondrial DNA, mtDNA, lineage back to this individual. Mitochondrial Eve is estimated to have lived between 150,000 and 200,000 years ago. Explanation. Mitochondrial Eve is identified by studying mitochondrial DNA, which is passed down from mothers to their offspring. Unlike nuclear DNA, which undergoes recombination and mixing, mitochondrial DNA is relatively stable and passes down with minimal changes, allowing scientists to trace the maternal lineage back to a common ancestor. Example. In a study published in 1987, researchers analyzed mtDNA from various populations and identified a common ancestor, estimated to have lived around 200,000 years ago, whom they named mitochondrial Eve. It's important to note that mitochondrial Eve is not the only woman alive at that time, but rather the only individual whose mtDNA line survived to the present day. Both Y-chromosome atom and mitochondrial Eve are important concepts in understanding human evolutionary history. They represent the points in our genetic lineage where the male and female lineages converge, indicating the common ancestry of all living humans. However, it's crucial to remember that Y-chromosome atom and mitochondrial Eve are not the only ancestors of humanity, but rather specific individuals whose genetic lineages have endured and passed down through generations. However, it's crucial to remember that Y-chromosome atom and mitochondrial Eve are not the only ancestors of humanity, but rather specific individuals whose genetic lineages have endured and passed down through generations. More points about this topics not mentioned points. ChatGPT. Certainly. Here are some additional points about Y-chromosome atom and mitochondrial Eve. Coalescence. The concept of Y-chromosome atom and mitochondrial Eve is based on the idea of coalescence. Coalescence refers to the point in time when the genetic lineages of all individuals in a population merge into a common ancestor. Y-chromosome atom and mitochondrial Eve represent the points of coalescence for their respective genetic lineages. Population size. The existence of Y-chromosome atom and mitochondrial Eve does not imply that they were the only individuals living at that time. They are simply the individuals from whom all existing Y-chromosomes and mitochondrial DNA can be traced. The population size at the time of Y-chromosome atom and mitochondrial Eve would have been much larger and would have included many other individuals. Incomplete lineage sorting. It's important to note that Y-chromosome atom and mitochondrial Eve lived at different times and in different populations. They are not a literal Adam and Eve pair. In fact, there would have been multiple males and females living at the same time as Y-chromosome atom and mitochondrial Eve who also contributed to the genetic diversity of modern humans. Mutation and genetic drift. Over time, mutations occur in the genetic material, including the Y-chromosome and mitochondrial DNA. This means that the genetic lineages of Y-chromosome atom and mitochondrial Eve would have evolved and diversified as new mutations accumulated and were passed down to subsequent generations. Additionally, genetic drift, which is the random fluctuation of allele frequencies in a population, would have influenced the genetic diversity and lineage distribution. Migration and interbreeding. The concepts of Y chromosome atom and mitochondrial Eve are based on the study of genetic lineages passed down through direct paternal or maternal inheritance. However, it's important to recognize that human populations have migrated and interbred throughout history. This means that individuals may carry genetic material from multiple ancestral lineages, and the concepts of Y chromosome atom and mitochondrial Eve represent only a subset of our overall genetic ancestry. Geographic considerations. The estimates for the time when Y chromosome atom and mitochondrial Eve lived are based on genetic analyses and assumptions about the rate of mutation accumulation. However, these estimates can vary depending on the specific population studied and the assumptions made. Furthermore, it's important to consider the geographical context, as different regions may have had separate lineages that eventually merged to form. The broader genetic diversity seen in modern humans. Limitations. While the concepts of Y chromosome atom and mitochondrial Eve provide insights into human genetic history, they have limitations. The analysis is based on the available genetic data, which may not capture the full complexity of human evolutionary history. Additionally, genetic studies cannot capture the full range of human diversity, including cultural, linguistic, and behavioral aspects. Overall, the concepts of Y chromosome atom and mitochondrial Eve provide a framework for understanding the common genetic ancestry of humans. They highlight the interconnectedness of our species and the shared evolutionary history that has shaped our genetic diversity. However, it's important to interpret these concepts in the context of ongoing research and our growing understanding of human evolutionary history. Can evolution explain language? In detail with examples and explanation in detail. ChatGPT. The question of whether evolution can explain language is a topic of ongoing scientific inquiry and debate. While there is no definitive answer, there are various theories and perspectives that shed light on the potential evolutionary origins of language. Here are some key points and explanations. Evolutionary adaptation. One perspective suggests that language may have evolved as an adaptation, providing humans with a selective advantage. Language allows for communication, social cooperation, and the transmission of knowledge across generations. By facilitating efficient communication, early humans could have coordinated their actions, shared information about food sources, warned of potential dangers, and formed complex social structures. Social and cultural evolution. Language is not solely a biological trait but also a product of social and cultural evolution. Humans possess a unique capacity for social learning, allowing us to acquire and transmit knowledge and behaviors through cultural means. Language likely emerged and evolved as a cultural phenomenon, with early humans developing shared systems of communication to enhance social cohesion and cooperation. Cognitive abilities. The evolution of language may be closely intertwined with the development of cognitive abilities in humans. Language requires complex mental processes such as syntax, grammar, and the ability to generate and comprehend meaningful sounds and symbols. Some argue that the cognitive capacities necessary for language emerged gradually, potentially driven by selective pressures for improved communication and problem-solving skills. Gestural and vocal communication. Before the emergence of fully developed language, early humans likely relied on a combination of gestural and vocal communication. Evidence from studies of non-human primates, such as chimpanzees and bonobos, suggests that these species use gestures and vocalizations to communicate and convey information. It is believed that early hominins also possessed such communication systems, which eventually evolved into more complex linguistic abilities. FOXP2 gene. The FOXP2 gene has been implicated in language development, as mutations in this gene can lead to language impairments. This gene is conserved across several species, including humans and other primates. It is thought to play a role in the neural development and coordination of the muscles involved in speech and language production. The presence of the FOXP2 gene in non-human species suggests that it predates the emergence of language and may have been co-opted during the evolution of language-related functions. Paleontological and archaeological evidence. The fossil record and archaeological findings provide indirect evidence related to the evolution of language. Brain imprints, endocranial casts, and archaeological artifacts associated with symbolic representation and communication, such as cave paintings, early musical instruments, and symbolic objects, provide hints about the cognitive abilities and cultural practices of early humans. It's important to note that the origins of language are still not fully understood, and there are various theories and hypotheses that continue to be explored. Language is a complex and multifaceted phenomenon, and its evolution likely involved a combination of genetic, cognitive, social, and cultural factors. While the precise mechanisms and timeline of language evolution remain a subject of ongoing research, the evidence suggests that language has played a significant role in shaping human societies and our unique cognitive abilities. More points. ChatGPT. Certainly. Here are some additional points regarding the potential connection between evolution and language. Genetic basis. Studies have identified genetic variations associated with language-related abilities. For example, the ASPM and MCPH1 genes have been implicated in brain development and cognition, including language acquisition. These genetic variations have undergone positive selection in humans, suggesting their potential role in the evolution of language-related traits. Comparative linguistics. Comparative studies of languages across different cultures and regions provide insights into the universal features of language and the potential evolutionary processes behind them. Linguists have identified shared grammatical structures, phonetic patterns, and semantic categories among diverse languages, suggesting a common evolutionary origin. FOXP2 in other species. While the FOXP2 gene has been extensively studied in humans, recent research has found variations in FOXP2 in other species, such as birds and mice. This indicates that the gene is involved in more general aspects of vocal communication and motor control, suggesting a broader evolutionary significance beyond human language. Tool use and symbolic behavior. The ability to use tools and engage in symbolic behavior is often considered a precursor to language. Evidence from archaeological sites shows the development of tool use and the creation of symbolic artifacts by early humans, indicating the presence of complex cognitive abilities related to problem-solving and communication. Neurological and brain imaging studies. Neuroscientific studies using brain imaging techniques have identified brain regions associated with language processing. Comparative studies involving humans and non-human primates have revealed differences in brain structures involved in language, providing insights into the evolutionary changes that have occurred in the human brain. Cultural evolution and language change. Language is a dynamic and evolving system that undergoes changes over time. Just as biological evolution occurs through the accumulation of genetic changes, language evolution can be seen as a cultural evolutionary process. Language change can result from various factors such as cultural contact, migration, and technological advancements. Paleolithic art. The presence of cave paintings and other forms of symbolic art in the Paleolithic era suggests the existence of sophisticated communication systems and the ability to convey abstract concepts. These artistic expressions provide indirect evidence of the early development of language and symbolic representation. Animal communication systems. Studying the communication systems of non-human animals, such as bees, birds, and dolphins, can provide insights into the evolutionary roots of language. While these systems differ from human language in complexity, they share some underlying principles of communication, such as the use of symbols and structured signals. It's important to note that the study of the evolution of language is a complex and interdisciplinary field, involving linguistics, biology, anthropology, and cognitive science. The points mentioned above represent different aspects and lines of evidence that contribute to our understanding of the potential evolutionary origins of language. Communication, speech, language and self. Awareness in primates. In detail with multiple examples and explanation in detail, each topic in detail 2 halves. ChatGPT. Certainly, let's explore each topic in detail. Communication in primates. Primates, including humans, have complex communication systems that involve various forms of signals, vocalizations, facial expressions, body postures, and gestures. These communication behaviors serve different purposes such as social bonding, mating, territory defense, and signaling danger. Here are some examples. Vocalizations. Primates produce a wide range of vocalizations, including calls, screams, hoots, and grunts. For instance, chimpanzees have distinct vocalizations for different situations, such as food calls or alarm calls. Gestures and body language. Primates use a variety of gestures and body movements to communicate intentions and emotions. For example, bonobos engage in open hand begging, extending their arms and reaching out to another individual as a request for food. Facial expressions. Primates, especially great apes, exhibit a rich repertoire of facial expressions that convey emotional states and social signals. For instance, a display of bared teeth in chimpanzees can indicate aggression or fear. Speech in primates. Speech refers specifically to the production of complex vocal sounds using the articulatory apparatus of the vocal tract. While non-human primates do not possess the anatomical capabilities for human-like speech, some studies have explored vocal learning abilities in certain species. For example, studies on non-human primates, such as orangutans and gorillas, have shown limited vocal learning abilities, where they can imitate and produce certain human-like sounds or even learn to use sign language. The studies on the vocal learning capabilities of birds, such as songbirds and parrots, have provided valuable insights into the neural mechanisms and evolutionary origins of vocal learning. Language in primates. Language, in the human context, involves the use of a complex and structured system of symbols, words, to convey meaning and express thoughts. While non-human primates do not possess full-fledged language abilities, researchers have explored their cognitive and communicative capacities. Examples include symbolic representations. Studies have shown that some primates, such as chimpanzees, can learn and use symbolic systems, including sign language or lexicograms, to communicate with humans. For instance, Kanzi, a bonobo, has been trained to use a lexigram board to communicate with humans and express desires or make requests. Comprehension and referential communication. Primates demonstrate an understanding of symbolic reference and can comprehend certain human words or signs. They can associate specific sounds or gestures with objects or actions in their environment. Self-awareness in primates. Self-awareness refers to the ability to recognize oneself as an individual entity separate from the environment and other individuals. Several primate species have shown signs of self-awareness, as demonstrated through the mirror self-recognition test. Notable examples include great apes, chimpanzees, bonobos, and orangutans have shown self-recognition abilities by recognizing their own reflections in mirrors and engaging in self-directed behaviors, such as examining body parts or using mirrors strategically. Dolphins and elephants. These non-primate species have also demonstrated self-awareness through mirror self-recognition tests, indicating a level of consciousness and self-perception. It is important to note that while primates exhibit various levels of communication, speech-like abilities, and aspects of language and self-awareness, they differ from human language and self-awareness in terms of complexity and linguistic productivity. Understanding these similarities and differences contributes to our knowledge of primate cognition and the evolution of communication and self-awareness in the animal kingdom. More details. ChatGPT. Certainly, here are more details on each topic. Communication in primates. Vocalizations. Primates use vocalizations to convey different messages. For example, certain vocalizations in vervet monkeys communicate specific predators, such as lovebird calls or eagle calls. Gestures and body language. Primates use a variety of gestures and body postures to communicate. For instance, gorillas beat their chests to display dominance, while bonobos engage in genital rubbing as a form of social bonding. Facial expressions. Facial expressions play a crucial role in primate communication. For instance, a bared teeth display in primates like baboons can convey aggression, while a relaxed face with open eyes indicates non-threatening behavior. Speech in primates. Vocal learning. While non-human primates do not possess the same vocal capabilities as humans, some species have shown limited vocal learning abilities. For example, studies have found that certain species of monkeys and apes can imitate human sounds or produce sounds resembling human speech. Laryngeal anatomy. The main anatomical difference between humans and non-human primates lies in the larynx. The position of the larynx in humans allows for precise control of vocalizations, whereas in non-human primates, it is situated higher, limiting their range of vocalizations. Language in primates. Symbolic representations. Primates can learn and use symbols to communicate. For example, the bonobo Kanzi was able to learn a lexigram system to communicate with humans, indicating the ability to associate symbols with meaning. Comprehension and referential communication. Primates have demonstrated an understanding of referential communication. They can learn to associate specific sounds or signs with objects or actions and respond accordingly. This suggests a level of comprehension and intentional communication. Self-awareness in primates. Mirror self-recognition. The mirror self-recognition test is a common method used to assess self-awareness in primates. It involves placing a mark on the animal's body and observing their reaction when they see themselves in a mirror. Primates that display self-awareness will inspect the mark on their own body rather than the mirror image. Levels of self-awareness. Different primate species exhibit varying levels of self-awareness. Great apes, such as chimpanzees and orangutans, generally show higher levels of self-awareness compared to other primates. Cognitive implications. Self-awareness in primates suggests a level of cognitive complexity and the ability to recognize oneself as an individual with thoughts, emotions, and intentions. Understanding communication, speech, language, and self-awareness in primates provides insights into the evolution of these abilities and helps us better understand the similarities and differences between humans and other primates in terms of cognitive and communicative capacities. Biostatistics. Biostatistics is the application of statistical methods to analyze and interpret data related to living organisms and their interactions with the environment. It is a branch of statistics that deals with the design and analysis of experiments in biology, medicine, public health, and related fields. Biostatistics is used to help researchers and healthcare professionals understand data and make informed decisions based on the data. Biostatistics can be used to analyze data from clinical trials, observational studies, epidemiological studies, and other types of studies. It can also be used to develop new statistical methods that are specifically designed to address the unique challenges of working with biological and medical data. Some common areas of biostatistics include designing and analyzing clinical trials to test new treatments or interventions, analyzing epidemiological data to understand the spread of diseases and risk factors, developing statistical models to understand the relationship between genetic and environmental factors in the development of disease, analyzing healthcare data to identify patterns and trends in healthcare utilization and outcomes, designing and analyzing studies to evaluate the effectiveness of public health interventions. Biostatistics plays a critical role in advancing our understanding of human health and disease and is essential for the development of new treatments and interventions. Next topic. Types of data in detail. In statistics, there are four main types of data: nominal, ordinal, interval, and ratio. Each type of data has different properties and characteristics that affect how it can be analyzed and interpreted. Nominal data. Nominal data is categorical data that represents names or labels. Nominal data cannot be ordered or ranked, and there is no inherent mathematical relationship between categories. Examples of nominal data include gender, race, eye color, and political affiliation. Ordinal data. Ordinal data is also categorical data, but it can be ordered or ranked. The categories in ordinal data have a natural order or sequence, but the differences between the categories are not necessarily equal. Examples of ordinal data include education level, e.g., high school, college, graduate school, socioeconomic status, e.g., low, middle, high, and movie ratings, e.g., poor, fair, good, excellent. Interval data. Interval data is numerical data that has a meaningful 0 point and equal intervals between values. Interval data does not have a true 0 point, meaning that a score of 0 does not indicate the absence of the variable being measured. Examples of interval data include temperature measured in Celsius or Fahrenheit, IQ scores, and dates on a calendar. Ratio data. Ratio data is also numerical data that has a meaningful 0 point and equal intervals between values, but unlike interval data, ratio data has a true 0 point that indicates the absence of the variable being measured. Ratio data can be used to calculate ratios and proportions. Examples of ratio data include height, weight, age, and income. Understanding the type of data being analyzed is essential for choosing appropriate statistical tests and interpreting results accurately. Nominal and ordinal data are often analyzed using non-parametric tests, while interval and ratio data are analyzed using parametric tests. Additionally, different types of data may require different types of graphical displays, such as bar charts for nominal and ordinal data and histograms or box plots for interval and ratio data. Next topic. Classification of data in the basis of source of data, compilation, variable, nature. Big essay. Classification of data. Data can be classified in various ways based on different criteria. In this essay, we will discuss the classification of data based on the source of data, compilation, variable, and nature. Based on the source of data. Data can be classified based on the source from where it is obtained. There are two main sources of data, primary and secondary. A. Primary data. Primary data is the data that is collected by the researcher from the original source for the first time. This data is raw and unprocessed and is collected through surveys, experiments, observations, and interviews. B. Secondary data. Secondary data is the data that is collected by someone else for some other purpose and is later used by the researcher. This data can be obtained from books, journals, government reports, websites, and other sources. Based on compilation. Data can be classified based on how it is compiled. There are two main types of data, individual and aggregate. A. Individual data. Individual data refers to data that is collected for each individual in a population or sample. This type of data is also known as raw data or unit data. B. Aggregate data. Aggregate data refers to data that is obtained by combining individual data. This type of data is also known as summary data or group data. Based on variable. Data can be classified based on the type of variable being measured. There are two main types of variables, qualitative and quantitative. A. Qualitative data. Qualitative data refers to data that is non-numeric and describes characteristics or attributes of the subject being studied. Examples of qualitative data include gender, race, religion, and marital status. B. Quantitative data. Quantitative data refers to data that is numeric and measures quantities or amounts. Examples of quantitative data include height, weight, temperature, and time. Based on nature. Data can be classified based on its nature. There are two main types of data, discrete and continuous. A. Discrete data. Discrete data refers to data that can only take on specific values and cannot be measured beyond those values. Examples of discrete data include the number of children in a family, the number of cars in a parking lot, and the number of students in a classroom. B. Continuous data. Continuous data refers to data that can take on any value within a range and can be measured to any degree of precision. Examples of continuous data include height, weight, and temperature. In conclusion, data can be classified based on various criteria such as the source of data, compilation, variable, and nature. Understanding the classification of data is important for selecting appropriate methods of data analysis and interpretation. Next topic. Methods of data collection and classification in detail with examples essay. Data collection is a crucial step in any research process. It involves gathering information or data from various sources that are relevant to the research question or problem. The methods of data collection can vary depending on the nature of the research question, the type of data needed, and the resources available. In this essay, we will discuss the methods of data collection and classification in detail with examples. Methods of data collection. Surveys. Surveys involve collecting data from a sample of individuals or groups through questionnaires or interviews. Surveys can be conducted through various modes such as mail, telephone, online, or in person. The data collected through surveys can be either qualitative or quantitative. Example, a company conducts a customer satisfaction survey to understand customer needs and improve their services. Observations. Observations involve collecting data by observing and recording the behavior or actions of individuals or groups in their natural setting. Observations can be either participant or non-participant, and the data collected can be either qualitative or quantitative. Example, a researcher observes children playing in a park to study their behavior and social interactions. Experiments. Experiments involve collecting data by manipulating a variable and observing the effect on the outcome variable. Experiments can be conducted in a laboratory or in the field, and the data collected is typically quantitative. Example, a researcher conducts an experiment to test the effectiveness of a new drug on patients with a specific medical condition. Case studies. Case studies involve collecting data by studying a particular individual, group, or situation in depth. The data collected through case studies can be either qualitative or quantitative. Example, a psychologist conducts a case study on a patient with a rare psychological disorder to understand the causes and symptoms of the disorder. Classification of data. Once data has been collected, it needs to be classified into different categories based on the type of data. The classification of data is important because it helps researchers to organize and analyze the data effectively. The following are the different types of data classifications. Quantitative data. Quantitative data is numerical data that can be measured and analyzed statistically. This type of data can be further classified as discrete or continuous. Example, the height and weight of students in a class are quantitative data. Qualitative data. Qualitative data is non-numerical data that describes a characteristic or attribute of the subject being studied. This type of data can be further classified as nominal or ordinal. Example, the gender and occupation of employees in a company are qualitative data. Primary data. Primary data is data that is collected by the researcher for the first time, and it can be further classified as raw or compiled data. Example, a survey conducted by a company to understand customer needs is an example of primary data. Secondary data. Secondary data is data that has already been collected by someone else for another purpose, and it can be further classified as published or unpublished data. Example, a researcher using data from a government report to conduct a study is an example of secondary data. In conclusion, the methods of data collection and classification are essential in any research process. Researchers need to carefully choose the appropriate method of data collection and classify the data accurately to ensure effective analysis and interpretation. 2 halves. Next topic. Types of sampling methods essay. Sampling is the process of selecting a subset of individuals or units from a larger population for the purpose of making inferences about the population as a whole. In research, sampling is crucial as it allows researchers to make inferences about a population without having to study every individual or unit within that population. In this essay, we will discuss the different types of sampling methods. Probability sampling. Probability sampling involves selecting individuals or units from a population randomly. This means that every individual or unit in the population has an equal chance of being selected for the sample. Probability sampling is considered the most reliable sampling method as it minimizes bias and provides a representative sample of the population. There are 4 types of probability sampling. I. Simple random sampling. In simple random sampling, individuals or units are selected randomly without any bias or preference. Example, a researcher selects 50 students from a college using a random number generator. E. Systematic sampling. In systematic sampling, individuals or units are selected at regular intervals from a list or sequence. Example, a researcher selects every 10th patient from a list of patients in a hospital. E. Stratified sampling. In stratified sampling, individuals or units are selected from specific subgroups or strata within the population. The subgroups are determined based on characteristics such as age, gender, or ethnicity. Example, a researcher selects a sample of 100 students from a college, with 25 students selected from each year group. IV. Cluster sampling. In cluster sampling, individuals or units are selected from groups or clusters within the population. The clusters are determined based on geographic location or other similar characteristics. Example, a researcher selects a sample of 50 households from a town by randomly selecting streets and then households within those streets. Non-probability sampling. Non-probability sampling involves selecting individuals or units from a population using non-random methods. This means that not all individuals or units in the population have an equal chance of being selected for the sample. Non-probability sampling is considered less reliable than probability sampling, as it can result in biased samples. There are 4 types of non-probability sampling. I. Convenience sampling. In convenience sampling, individuals or units are selected based on their availability and accessibility. Example, a researcher selects participants for a study by approaching people on the street. E. Quota sampling. In quota sampling, individuals or units are selected based on predetermined quotas for certain characteristics such as age or gender. Example, a researcher selects 50 participants for a study, with 25 males and 25 females. E. Purpose of sampling. In purpose of sampling, individuals or units are selected based on specific criteria determined by the researcher. Example, a researcher selects participants for a study based on their expertise in a particular field. IV. Snowball sampling. In snowball sampling, individuals or units are selected based on referrals from other participants. Example, a researcher selects participants for a study by asking current participants to refer other potential participants. In conclusion, the choice of sampling method depends on the research question, the nature of the population, and the available resources. Probability sampling is generally considered more reliable than non-probability sampling, but non-probability sampling can be useful in certain situations where probability sampling is not feasible. It is important for researchers to carefully consider their sampling method to ensure that their sample is representative of the population and minimizes bias. Next topic. Advantages and disadvantages of census and sampling methods in detail essay. In research, there are 2 primary methods of collecting data, census and sampling. Census involves collecting data from every individual or unit within a population, while sampling involves selecting a subset of individuals or units from a population for the purpose of making inferences about the population as a whole. In this essay, we will discuss the advantages and disadvantages of census and sampling methods. Advantages of census. Accurate representation of the population. A census provides a complete and accurate representation of the population, as every individual or unit is included in the data collection process. Detailed information. A census provides detailed information about the population, including demographic data, socioeconomic data, and other relevant variables. Useful for small populations. A census is useful for small populations where it is feasible to collect data from every individual or unit. Disadvantages of census. Expensive. Conducting a census can be expensive, as it requires significant resources and time to collect data from every individual or unit within a population. Time-consuming. A census can be time-consuming, as it requires data to be collected from every individual or unit within a population. Privacy concerns. A census may raise privacy concerns, as individuals may be uncomfortable with sharing personal information. Advantages of sampling. Cost-effective. Sampling is a cost-effective method of data collection, as it requires fewer resources and less time than a census. Time-efficient. Sampling is a time-efficient method of data collection, as it only requires data to be collected from a subset of individuals or units within a population. Representative sample. Sampling can provide a representative sample of a population, as long as the sampling method is appropriate and unbiased. Disadvantages of sampling. Risk of bias. Sampling can be subject to bias, particularly if the sampling method is not appropriate or if there is a sampling error. Less detailed information. Sampling provides less detailed information than a census, as only a subset of individuals or units are included in the data collection process. Limited use for small populations. Sampling is less useful for small populations, as the sample size may be too small to provide a representative sample. In conclusion, both census and sampling methods have advantages and disadvantages, and the choice of method depends on the research question, the nature of the population, and the available resources. A census is generally more accurate and detailed, but it can be expensive and time-consuming. Sampling is more cost-effective and time-efficient, but it can be subject to bias and provide less detailed information. Researchers should carefully consider their method of data collection to ensure that it is appropriate for their research question and population. Next topic. Class intervals. Exclusive and inclusive methods in detail with examples. Essay in statistical analysis. Class intervals are used to group data into intervals or ranges. These intervals are used in histograms, frequency tables, and other statistical analyses. There are two methods for defining class intervals, exclusive and inclusive. In this essay, we will discuss these two methods in detail, along with examples. Exclusive class intervals. In the exclusive method, the upper limit of one interval is not included in the lower limit of the next interval. For example, if we have a set of data ranging from 0 to 100 and we want to create 5 class intervals, we can use the following exclusive intervals. 0 to 19. 20 to 39. 40 to 59. 60 to 79. 80 to 99. In this example, the upper limit of each interval is not included in the lower limit of the next interval. This method is commonly used in statistics and is preferred when the data is continuous. Advantages of exclusive class intervals. No overlap. Exclusive class intervals do not overlap, which makes it easy to determine which interval a value falls into. Useful for continuous data. Exclusive class intervals are useful for continuous data, as they allow for a clear and accurate representation of the data. Disadvantages of exclusive class intervals. Limitations for discrete data. Exclusive class intervals may not be suitable for discrete data, as there may be gaps between intervals. Ambiguity. There may be ambiguity at the endpoints of the intervals, as it may not be clear whether a value belongs to one interval or the next. Inclusive class intervals. In the inclusive method, the upper limit of one interval is included in the lower limit of the next interval. For example, if we have a set of data ranging from 0 to 100 and we want to create 5 class intervals, we can use the following inclusive intervals. 020 21 40 41 60 61 80. 81 to 100. In this example, the upper limit of each interval is included in the lower limit of the next interval. This method is less commonly used in statistics, but it is preferred when dealing with discrete data. Advantages of inclusive class intervals. Suitable for discrete data. Inclusive class intervals are suitable for discrete data, as they do not create gaps between intervals. No ambiguity. There is no ambiguity at the endpoints of the intervals, as values that fall on the boundary are included in the interval. Disadvantages of inclusive class intervals. Overlap. Inclusive class intervals overlap, which can make it difficult to determine which interval a value belongs to. Unsuitable for continuous data. Inclusive class intervals may not be suitable for continuous data, as the overlap between intervals can distort the representation of the data. In conclusion, both exclusive and inclusive class interval methods have advantages and disadvantages, and the choice of method depends on the nature of the data and the analysis being performed. Exclusive class intervals are preferred for continuous data, while inclusive class intervals are suitable for discrete data. It is important to carefully consider the choice of class interval method to ensure that it accurately represents the data and provides meaningful insights. 2 halves. Next topic. Frequency curve essay. In statistics, a frequency curve, also known as a density curve, is a graphical representation of a frequency distribution. It shows the distribution of data in a continuous or interval scale in a smooth curve. In this essay, we will discuss frequency curves in detail, including their types, characteristics, and how they are used in statistical analysis. Types of frequency curves. There are several types of frequency curves, including the normal curve, skewed curve, bimodal curve, and uniform curve. Normal curve. The normal curve is a bell-shaped curve that is symmetrical around the mean. It represents a normal distribution of data, where the majority of the data falls around the mean, and the extreme values are rare. This type of curve is commonly used in statistics, especially in hypothesis testing. Skewed curve. A skewed curve is a frequency curve that is not symmetrical, and the data is distributed more towards one end than the other. There are 2 types of skewed curves, left skewed and right skewed. Bimodal curve. A bimodal curve is a frequency curve that has 2 distinct peaks, which indicates that there are 2 different groups of data in the distribution. Uniform curve. A uniform curve is a frequency curve where the data is evenly distributed across the entire range, and there is no prominent peak or trough. Characteristics of frequency curves. Frequency curves have several important characteristics, including shape. The shape of the frequency curve indicates the type of distribution of the data. Symmetry. The symmetry of the frequency curve indicates the balance between the values on either side of the mean. Peak. The peak of the frequency curve indicates the value or range of values that occur most frequently in the distribution. Spread. The spread of the frequency curve indicates how widely dispersed the data is across the range. Uses of frequency curves. Frequency curves are used in statistical analysis to provide a visual representation of the distribution of data. They are used to determine the shape and characteristics of a distribution, identify outliers or extreme values in the distribution, compare different distributions and determine if they are similar or different, calculate the probability of specific values occurring in the distribution. In conclusion, frequency curves are a powerful tool for understanding and analyzing data in statistics. They provide a visual representation of the distribution of data, and their shape, symmetry, peak, and spread can reveal important insights about the data. Understanding the characteristics of frequency curves is essential for conducting accurate statistical analysis and drawing meaningful conclusions. Next topic. Frequency curve types, skewness, kurtosis, ogive in detail with examples essay. Frequency curve, also known as density curve, is a graphical representation of the frequency distribution of a dataset. It shows the distribution of data in a continuous or interval scale in a smooth curve. In this essay, we will discuss in detail the types of frequency curves, skewness, kurtosis, and ogive. Types of frequency curves. Normal curve. The normal curve is a bell-shaped curve that is symmetrical around the mean. It represents a normal distribution of data, where the majority of the data falls around the mean, and the extreme values are rare. This type of curve is commonly used in statistics, especially in hypothesis testing. Skewed curve. A skewed curve is a frequency curve that is not symmetrical, and the data is distributed more towards one end than the other. There are two types of skewed curves, left skewed and right skewed. In a left skewed curve, the tail of the curve is on the left, and in a right skewed curve, the tail is on the right. Bimodal curve. A bimodal curve is a frequency curve that has two distinct peaks, which indicates that there are two different groups of data in the distribution. Uniform curve. A uniform curve is a frequency curve where the data is evenly distributed across the entire range, and there is no prominent peak or trough. Skewness. Skewness is a measure of the asymmetry of a frequency distribution. A distribution can be positively skewed, negatively skewed, or symmetrical. A positively skewed distribution means that the tail of the distribution is longer on the right side, and the mean is greater than the median. A negatively skewed distribution means that the tail of the distribution is longer on the left side, and the mean is less than the median. Kurtosis. Kurtosis is a measure of the peakness of a frequency distribution. A distribution can be leptokurtic, mesokurtic, or platykurtic. Leptokurtic distributions are more peaked than the normal distribution, mesokurtic distributions are similar to the normal distribution, and platykurtic distributions are flatter than the normal distribution. Ogive. Ogive is a graph that shows the cumulative frequency distribution of a dataset. It is used to visualize the cumulative frequency distribution and to determine the percentage of data that falls below a certain value. There are two types of ogives, the less than ogive, which shows the cumulative frequency of data that is less than or equal to a certain value, and the greater than ogive, which shows the cumulative frequency of data that is greater than or equal to a certain value. Examples. Suppose we have a dataset of the weights of a sample of individuals. The frequency distribution of this dataset can be represented by a frequency curve. The type of frequency curve will depend on the distribution of the data. If the weights follow a normal distribution, the frequency curve will be bell-shaped, symmetrical, and the mean and median will be equal. If the weights are positively skewed, the frequency curve will have a longer tail on the right side, and the mean will be greater than the median. If the weights are negatively skewed, the frequency curve will have a longer tail on the left side, and the mean will be less than the median. If the weights are uniformly distributed, the frequency curve will be flat, and the data will be evenly distributed across the range. An ogive can be used to determine the percentage of individuals whose weight is below or above a certain value. In conclusion, frequency curves, skewness, kurtosis, and ogives are important tools in statistical analysis. They provide a visual representation of the distribution of data and help to understand the characteristics of the dataset. Next topic. Statistical methods, measures of central tendency and dispersion essay. In statistical analysis, measures of central tendency and dispersion are important tools to describe the characteristics of a dataset. In this essay, we will discuss in detail the measures of central tendency and dispersion. Measures of central tendency. Measures of central tendency are used to describe the central value of a dataset. There are three types of measures of central tendency. Mean. The mean is the arithmetic average of the dataset. It is calculated by adding up all the values in the dataset and dividing by the number of values. Median. The median is the middle value of the dataset when the values are arranged in order. If there is an even number of values, the median is the average of the two middle values. Mode. The mode is the value that occurs most frequently in the dataset. The mean is the most commonly used measure of central tendency. However, if the dataset contains extreme values or outliers, the median may be a better measure of central tendency as it is less affected by outliers. Measures of dispersion. Measures of dispersion are used to describe the spread of a dataset. There are three types of measures of dispersion. Range. The range is the difference between the highest and lowest values in the dataset. Variance. The variance measures how far each value in the dataset is from the mean. It is calculated by taking the sum of the squared differences between each value and the mean, and then dividing by the number of values. Standard deviation. The standard deviation is the square root of the variance. It measures the average distance of each value from the mean. The standard deviation is the most commonly used measure of dispersion. It is useful in determining the spread of the dataset, as well as identifying outliers. A larger standard deviation indicates a larger spread of the data, while a smaller standard deviation indicates a smaller spread of the data. Examples. Suppose we have a dataset of the heights of a sample of individuals. We can calculate the measures of central tendency and dispersion to describe the characteristics of the dataset. The mean height can be calculated by adding up all the values and dividing by the number of values. The median height can be found by arranging the values in order and finding the middle value. The mode height is the value that occurs most frequently. To calculate the range, we find the difference between the highest and lowest values. The variance can be calculated by taking the sum of the squared differences between each value and the mean, and dividing by the number of values. The standard deviation is the square root of the variance. In conclusion, measures of central tendency and dispersion are important tools in statistical analysis. They provide a summary of the dataset and help to understand the characteristics of the data. The choice of measure depends on the nature of the data and the purpose of the analysis. Next topic. Mean essay raw data, discrete series and continuous series. How to calculate with examples. Mean is a measure of central tendency used to describe the average of a dataset. There are different methods of calculating the mean depending on the type of data. In this essay, we will discuss how to calculate the mean for raw data, discrete series, and continuous series. Mean for raw data. Raw data refers to a set of individual values. To calculate the mean of raw data, we need to add up all the values and divide by the number of values in the dataset. For example, suppose we have a dataset of the weights of 6 people, 70 kilograms, 75 kilograms, 68 kilograms, 72 kilograms, 80 kilograms, and 65 kilograms. To calculate the mean weight, we add up all the weights and divide by the number of people. Mean weight equals 70 plus 75 plus 68 plus 72 plus 80 plus 65, 6 equals 70 kilogram. Therefore, the mean weight of these 6 people is 70 kilogram. Mean for discrete series. Discrete series refers to a set of values that can only take on certain values, such as integers. To calculate the mean for discrete series, we need to multiply each value by its frequency, add up the products, and divide by the total frequency. For example, suppose we have a dataset of the number of books read by a group of students. Number of books read frequency. 02. 13. 26. 34. 42. To calculate the mean number of books read, we multiply each value by its frequency, add up the products, and divide by the total frequency. Mean number of books read equals 0 times 2 plus 1 times 3 plus 2 times 6 plus 3 times 4 plus 4 times 2, 17 equals 1.94. Therefore, the mean number of books read by these students is 1.94. Mean for continuous series. Continuous series refers to a set of values that can take on any value within a range. To calculate the mean for continuous series, we need to find the midpoint of each class interval, multiply it by its frequency, add up the products, and divide by the total frequency. For example, suppose we have a dataset of the heights of a group of people. Height in cm, frequency 150-1605, 160-1708, 170-180-12, 180-1906, 190-202. To calculate the mean height, we need to find the midpoint of each class interval. The midpoint of the first interval is 150 plus 160, 2 equals 155, and so on. Then, we multiply each midpoint by its frequency, add up the products, and divide by the total frequency. Mean height equals 155 times 5 plus 165 times 8 plus 175 times 12 plus 185 times 6 plus 195 times 2. Discrete series, we multiply each value by its frequency, add up the products, and divide by the total frequency. In a series, we find the midpoint of each class interval, multiply it by its frequency, add up the products, and divide by the total frequency. Calculating standard deviation. Standard deviation essay raw data. How to calculate with examples. Standard deviation is a measure of the variability or spread of a set of data. It tells us how much the values in a dataset are spread out from the mean average value. Calculating standard deviation involves several steps. Calculate the mean average of the dataset. For each value in the dataset, subtract the mean from the value to get the deviation. Square each deviation value. Add up all the squared deviation values. Divide the sum of squared deviations by the total number of values in the dataset minus 1. Take the square root of the result from step 5 to get the standard deviation. Here's an example to illustrate how to calculate standard deviation. Let's say we have the following dataset: 5, 6, 7, 8, 9. Step 1. Calculate the mean. Mean equals 5 plus 6 plus 7 plus 8 plus 9. 5 equals 7. Step 2. Calculate the deviation for each value in the dataset. Deviation of 5 equals 5. 7 equals minus 2. Deviation of 6 equals 6. 7 equals minus 1. Deviation of 7 equals 7. 7 equals 0. Deviation of 8 equals 8. 7 equals 1. Deviation of 9 equals 9. 7 equals 2. Step 3. Square each deviation value. Minus 2 squared equals 4. Minus 1 squared equals 1. 0 squared equals 0. 1 squared equals 1. 2 squared equals 4. Step 4. Add up all the squared deviation values. 4 plus 1 plus 0 plus 1 plus 4 equals 10. Step 5. Divide the sum of squared deviations by the total number of values minus 1. 10 quarters equals 2.5. Step 6. Take the square root of the result from step 5 to get the standard deviation. SQRT 2.5 equals 1.58. Therefore, the standard deviation of the dataset is approximately 1.58. This tells us that the values in the dataset are relatively close to the mean 7 with only a moderate amount of variability. Standard deviations essay, discrete series and how to calculate with examples. Standard deviation is a statistical measure used to quantify the amount of dispersion or variability in a set of data. It is a measure of the spread of the data around the mean value. When dealing with a discrete series of data, which consists of a finite set of values with no intermediate values, the formula for calculating the standard deviation is slightly different from that used for a continuous series of data. To calculate the standard deviation of a discrete series of data, you can use the following formula: S equals SQRT sigma xx squared n1, where S is the standard deviation, x is the value in the dataset, x is the mean value of the dataset, and n is the total number of values in the dataset. Here's an example to illustrate how to calculate the standard deviation of a discrete series of data. Suppose you have a dataset of exam scores for a class of 10 students: 80, 75, 90, 85, 95, 70, 65, 78, 88, 92. Step 1. Calculate the mean of the dataset. X equals 80 plus 75 plus 90 plus 85 plus 95 plus 70 plus 65 plus 78 plus 88 plus 92. 10 equals 82. Step 2. Calculate the deviation for each value in the dataset. Deviation of 80 equals 80. 82 equals 2. Deviation of 75 equals 75. 82 equals minus 7. Deviation of 90 equals 90. 82 equals 8. Deviation of 85 equals 85. 82 equals 3. Deviation of 95 equals 95. 82 equals 13. Deviation of 70 equals 70. 82 equals minus 12. Deviation of 65 equals 65. 82 equals minus 17. Deviation of 78 equals 78. 82 equals minus 4. Deviation of 88 equals 88. 82 equals 6. Deviation of 92 equals 92. 82 equals 10. Step 3. Square each deviation value. Minus 2 squared equals 4. Minus 7 squared equals 49. 8 squared equals 64. 3 squared equals 9. 13 squared equals 169. Minus 12 squared equals 144. Minus 17 squared equals 289. Minus 4 squared equals 16. 6 squared equals 36. 10 squared equals 100. Step 4. Add up all the squared deviation values. Sigma xx squared equals 4 plus 49 plus 64 plus 9 plus 169 plus 144 plus 289 plus 16 plus 36 plus 100 equals 880. Step 5. Divide the sum of squared deviations by the total number of values minus 1. S squared equals sigma xx squared n1 equals 880. 10 equals 97.78. Step 6. Take the square root of the result from step 5 to get the standard deviation. S equals SQRT S squared equals SQRT 97.78 equals 9.89. Therefore, the standard deviation of the exam scores for this class is approximately 9.89. This tells us that the scores are spread out from the mean value of 82 by approximately 9.89 points. The larger the standard deviation, the more spread out the data is. The smaller the standard deviation, the more clustered the data is around the mean value. Standard deviation essay continuous series. How to calculate with examples in statistics. A continuous series of data is one in which the values are not restricted to a specific set of discrete values, but rather can take on any value within a range. For example, the height of a group of people or the weight of a sample of products are continuous variables. To calculate the standard deviation of a continuous series of data, you can use the following formula: S equals SQRT sigma xx squared n1, where S is the standard deviation, x is the value in the dataset, x is the mean value of the dataset, and n is the total number of values in the dataset. However, in the case of continuous data, the formula becomes more complex since the range of possible values is infinite. Therefore, instead of calculating the standard deviation for each individual value, we need to calculate the area under the curve of the probability density function of the distribution. One common example of a continuous distribution is the normal distribution. The normal distribution is a bell-shaped curve that describes the probability of occurrence of a random variable. It is characterized by two parameters, the mean, mu, and the standard deviation, sigma. The formula for the standard deviation of a normal distribution is S equals sigma equals SQRT, sigma x mu squared n, where S is the standard deviation, sigma is the population standard deviation, x is the value in the dataset, mu is the mean value of the dataset, and n is the total number of values in the dataset. Here's an example to illustrate how to calculate the standard deviation of a continuous series of data using the normal distribution. Suppose you have a dataset of the height of a group of 100 people, and the mean height is 170 centimeters with a standard deviation of 10 centimeters. Step 1. Identify the mean and the standard deviation of the dataset. Mu equals 170. Sigma equals 10. Step 2. Use the formula to calculate the standard deviation. S equals sigma equals 10. In this case, the standard deviation of the dataset is equal to the population standard deviation, which is 10 centimeters. This means that the height of the people in the dataset is relatively tightly clustered around the mean value of 170 centimeters, with most of the people falling within one standard deviation of the mean, which is between 160 centimeters and 180 centimeters. In summary, calculating the standard deviation for a continuous series of data involves determining the probability density function of the distribution and calculating the area under the curve. For a normal distribution, the formula is simplified, as shown above, but for other types of distributions, more complex calculations may be required. Calculation of standard error. Next topic. Standard error SA raw data, discrete series and continuous series. How to calculate with examples. Standard error is a statistical measure that indicates the variability of a sample mean estimate from the true population mean. It is used to estimate the precision of a sample mean as an estimate of the true population mean. The formula for calculating the standard error depends on the type of data you have, raw data, discrete series, or continuous series. Standard error for raw data. When you have raw data, the standard error can be calculated using the following formula. SE equals S SQRT n. Where SE is the standard error, S is the sample standard deviation, and n is the sample size. For example, suppose you have a sample of 10 test scores, 75, 80, 85, 90, 85, 95, 90, 85, 80, and 90. The sample mean is 86 and the sample standard deviation is 5.56. The standard error can be calculated as SE equals 5.56 SQRT 10 equals 1.76. Therefore, the standard error for this sample is 1.76. Standard error for discrete series. When you have discrete series data, the standard error can be calculated using the following formula. SE equals SQRT sigma f xx squared n1 SQRT n. Where SE is the standard error, f is the frequency of each value, x is the value, x is the sample mean, and n is the sample size. For example, suppose you have a sample of 20 heights in inches, 62, 64, 66, 68, 70, 72, 74, and 76, each occurring twice. The sample mean is 68 and the standard deviation is 4.14. The standard error can be calculated as SE equals SQRT sigma f xx squared n1 SQRT n equals SQRT 2 62 to 68 squared plus 2 64 to 68 squared plus 2 76 to 68 squared 21 SQRT 20 equals 0.92. Therefore, the standard error for this sample is 0.92. Standard error for continuous series. When you have continuous series data, the standard error can be calculated using the following formula. SE equals S SQRT n. Where SE is the standard error, S is the sample standard deviation, and n is the sample size. For example, suppose you have a sample of 50 weights of apples in grams, ranging from 100 to 200 grams. The sample mean is 150 grams and the sample standard deviation is 20 grams. The standard error can be calculated as SE equals 20 SQRT 50 equals 2.83. Therefore, the standard error for this sample is 2.83. In summary, the standard error is a useful statistic for estimating the precision of a sample mean as an estimate of the true population mean. The formula for calculating the standard error depends on the type of data you have, raw data, discrete series, or continuous series. Next topic. Degree of freedom SA raw data, discrete series and continuous series. How to calculate with examples. Degrees of freedom DF is a concept used in statistics to describe the number of independent pieces of information in a sample or dataset. It is a crucial concept when calculating statistical tests such as t-tests, chi-square tests, and ANOVA. The degree of freedom depends on the type of data you have, raw data, discrete series, or continuous series. Degree of freedom for raw data. When you have raw data, the degree of freedom can be calculated as df equals n1. Where df is the degree of freedom and n is the sample size. For example, suppose you have a sample of 10 test scores, 75, 80, 85, 90, 85, 95, 90, 85, 80, and 90. The degree of freedom can be calculated as df equals 10 1 equals 9. Therefore, the degree of freedom for this sample is 9. Degree of freedom for discrete series. When you have discrete series data, the degree of freedom can be calculated as df equals nk. Where df is the degree of freedom, n is the sample size, and k is the number of distinct values in the sample. For example, suppose you have a sample of 20 heights in inches, 62, 64, 66, 68, 70, 72, 74, and 76, each occurring twice. The degree of freedom can be calculated as k equals 8, n equals 20, df equals nk, equals 28, equals 12. Therefore, the degree of freedom for this sample is 12. Degree of freedom for continuous series. When you have continuous series data, the degree of freedom can be calculated as df equals n1. Where df is the degree of freedom and n is the sample size. For example, suppose you have a sample of 50 weights of apples in grams, ranging from 100 to 200 grams. The degree of freedom can be calculated as df equals 51 equals 49. Therefore, the degree of freedom for this sample is 49. In summary, the degree of freedom is a crucial concept in statistics used to calculate statistical tests such as t-tests, chi-square tests, and ANOVA. The degree of freedom depends on the type of data you have, raw data, discrete series, or continuous series. The formulas for calculating the degree of freedom for each type of data are different and should be used accordingly. Open sidebar. Quartile deviation and box plot. Next topic. Quartile deviation SA box whisker plot essay with example and steps and importance. The quartile deviation and box and whisker plots are two tools commonly used in statistics to visualize and analyze datasets. In this essay, we will discuss what these tools are, how they are calculated, and their importance in understanding and interpreting data. Firstly, the quartile deviation is a measure of dispersion that indicates how spread out a dataset is by measuring the distance between the first and third quartiles. The first quartile Q1 is the value below which 25% of the data falls, while the third quartile Q3 is the value below which 75% of the data falls. The quartile deviation QD is calculated as half the difference between Q3 and Q1. QD equals Q3 Q1 2. For example, let's consider a dataset of 10 values, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. The first quartile Q1 is 3, the third quartile Q3 is 8, and the quartile deviation QD is 8. 3, 2 equals 2.5. This means that 50% of the data falls within 2.5 units of the median, which is the value that divides the dataset in half. The box and whisker plot is a graphical representation of a dataset that displays the quartiles, quartile deviation, and outliers, if any. The plot consists of a box that represents the middle 50% of the data, with a vertical line inside the box that represents the median. The whiskers extend from the box to the smallest and largest values that are not considered outliers. Outliers are represented as individual data points outside the whiskers. Let's use the same dataset as above to construct a box and whisker plot. First, we need to calculate the quartiles and quartile deviation, which we already did. The box will extend from Q1 to Q3, which are 3 and 8, respectively. The median is 5.5, which falls in the middle of the box. The whiskers will extend from 1 to 2.5 and from 8.5 to 10. These values are beyond the quartiles and quartile deviation and therefore considered outliers. The importance of the quartile deviation and box and whisker plot lies in their ability to summarize and communicate key features of a dataset, such as its central tendency, dispersion, and outliers. By visualizing the data in this way, it is easier to identify patterns, trends, and anomalies that may be important for decision-making. Additionally, these tools provide a standardized way to compare multiple datasets and draw meaningful conclusions from them. In conclusion, the quartile deviation and box and whisker plot are important tools in statistics for summarizing and visualizing data. The quartile deviation measures dispersion, while the box and whisker plot displays the quartiles, quartile deviation, and outliers. These tools can help to identify patterns, trends, and anomalies in datasets and make informed decisions based on them. Next topic. Basic concepts of probability and definition. Probability is a branch of mathematics that deals with the study of random events and their likelihood of occurrence. The basic concept of probability involves the measurement of the chances of a specific event happening or not happening, expressed as a fraction or percentage. Probability is defined as the ratio of the number of favorable outcomes to the total number of possible outcomes in a given event. It can be represented by the formula, probability equals number of favorable outcomes, total number of possible outcomes. For example, consider a fair coin toss. The possible outcomes are either heads or tails. The probability of getting heads is 1/2, as there is one favorable outcome, heads, out of two possible outcomes, heads or tails. Similarly, the probability of getting tails is also 1/2. Probability can range from 0 to 1, where 0 represents an event that is impossible, and 1 represents an event that is certain to happen. An event with a probability of 0.5 is equally likely to happen or not happen. There are two types of probabilities, theoretical probability and experimental probability. Theoretical probability is a probability calculated based on mathematical principles and assumptions, while experimental probability is a probability calculated based on actual experiments or observations. In conclusion, probability is a measure of the likelihood of an event occurring. It is calculated by dividing the number of favorable outcomes by the total number of possible outcomes. Probability can range from 0 to 1 and is used in a wide range of fields, including science, engineering, finance, and sports. Laws of probability essay. The laws of probability are fundamental principles that govern the likelihood of events occurring. They are essential to the study of statistics, and have numerous applications in fields such as finance, engineering, and science. In this essay, we will discuss the 3 fundamental laws of probability and their applications. The law of large numbers. This law states that as the number of trials in a probability experiment increases, the experimental probability of an event approaches its theoretical probability. In other words, if we perform an experiment many times, the average result will be close to the expected result. This law is the basis for many statistical techniques, including hypothesis testing and confidence intervals. For example, consider the experiment of tossing a fair coin. The theoretical probability of getting heads is 1/2. If we toss the coin 10 times, we might get 6 heads and 4 tails. However, if we toss the coin 100 times, we are more likely to get a result closer to the theoretical probability of 1/2, such as 48 heads and 52 tails. The addition law. This law states that the probability of the union of two events is equal to the sum of their individual probabilities, minus the probability of their intersection. In other words, if we have two events A and B, the probability of either event happening is the sum of their probabilities, minus the probability of both events happening together. For example, consider rolling a standard 6-sided die. Laws of probability essay. The laws of probability are fundamental principles that govern the likelihood of events occurring. They are essential to the study of statistics, and have numerous applications in fields such as finance, engineering, and science. In this essay, we will discuss the 3 fundamental laws of probability and their applications. The law of large numbers. This law states that as the number of trials in a probability experiment increases, the experimental probability of an event approaches its theoretical probability. In other words, if we perform an experiment many times, the average result will be close to the expected result. This law is the basis for many statistical techniques, including hypothesis testing and confidence intervals. For example, consider the experiment of tossing a fair coin. The theoretical probability of getting heads is 1/2. If we toss the coin 10 times, we might get 6 heads and 4 tails. However, if we toss the coin 100 times, we are more likely to get a result closer to the theoretical probability of 1/2, such as 48 heads and 52 tails. The addition law. This law states that the probability of the union of two events is equal to the sum of their individual probabilities, minus the probability of their intersection. In other words, if we have two events A and B, the probability of either event happening is the sum of their probabilities, minus the probability of both events happening together. For example, consider rolling a standard 6-sided die. The probability of rolling a 2 is 1/6, and the probability of rolling an even number is 3/6, since 3 of the 6 possible outcomes are even. The probability of rolling either a 2 or an even number is 4/6, which is the sum of the probabilities of the individual events minus the probability of both events happening together, which is 1/6, since rolling a 2 is not an even number. The multiplication law. This law states that the probability of the intersection of two events is equal to the product of their individual probabilities, given that the events are independent. In other words, if we have two events n b that are independent, the probability of both events happening together is equal to the product of their individual probabilities. For example, consider flipping a fair coin twice. The probability of getting heads on the first flip is 1/2, and the probability of getting heads on the second flip is also 1/2. The probability of getting heads on both flips, i.e., the intersection of the two events, is 1/2 by 1/2 equals 1/4. In conclusion, the laws of probability are fundamental principles